Welcome, everybody. We'll get started in about another minute or so. Well, welcome to our April. We have two speakers in April for this month, our Dean Seminar, as we sort of wrap up the semester. It is amazing to think that we're almost there at the end of the semester. I have the great pleasure of introducing our Associate Dean for Undergraduate Studies and Associate Professor, Dr. Laura Vandenberg from the Department of Environmental Health Sciences. For those of you who don't know, Laura is actually not a public health trained scientist. She got her undergraduate, her doctoral degree in molecular and developmental biology from Tufts University in their School of Medicine, and has done several postdocs in this area of regenerative and developmental biology. And yet when you hear her speak, when you hear her teach, she is amazing because she has this great depth of understanding of public health. And we are in for a delightful talk today, titled From Mice to Women, How Environmental Chemicals Disrupt Maternal and Infant Health and What We Can Do About It. So Laura, thank you so much for agreeing to speak at the Dean Seminar, and we look forward to hearing what you have to say.
Great, thank you. I'm going to start just by pointing out this adorable vervet monkey pair, maternal infant pair. This is a picture I took in South Africa, and these monkeys are the naughtiest monkeys, but they also really have amazing infant maternal bonds, and going to see them and how they interact with each other is just one of the most delightful things that I've had the opportunity to do on some of my science travels. I would like to start by telling you a story. And this is the story, really a very famous story from endocrinology and at the intersection of toxicology. It's the story of a drug called diethylstilbestrol, or DES. And DES was a drug that promised a whole bunch. It was given to somewhere around 10 million pregnant women in the US alone between the 1940s and the early 1970s. And it was given to women under the mistaken notion that if some estrogen during pregnancy is good for you, then more must be better. So it's a synthetic estrogen that was prescribed to women to prevent a spontaneous miscarriage. It promised women bigger and stronger babies too. This is an ad from the 1950s when there was no restrictions on what you could say in an advertisement. So this turns out actually not to be true. And while it was originally prescribed to women who were at risk for a miscarriage because they'd had one previously, it ultimately was recommended for routine prophylaxis, meaning okay for any woman to take, and actually recommended for any woman to take. Now, what happened in 1971 is that 6 young girls were seen in the office of Dr. Albert Herbst at Massachusetts General Hospital, and they had a very rare cancer called clear cell adenocarcinoma of the vagina. Not only is this a very rare cancer, it's normally diagnosed in elderly women. And so the story goes, one of the mothers says to Dr. Herbst, "Is this because of that drug that I took when I was pregnant?" And he says, "Probably not," but he starts to ask all of the mothers, and in fact, they had all taken DES during their pregnancy. And so this is the first real case study that documents that what has happened to these young girls in the womb with hormones, with excessive hormone exposure, would contribute to a disease that would manifest when they reach the age of puberty. DES, after Dr. Herbst published that result, was banned in the United States, but continued to be used in other countries through the 1980s. Now, DES was also prescribed to women as birth control, And it was prescribed to women as a way to stop lactation. So women who did not want to lactate could take DES shortly after giving birth as a way to cause their milk to dry up. So DES from a very early point was recognized to have an effect on breast health. In the 1970s, when it was found that DES caused this very rare cancer of the vagina, researchers went to the rodent to try to see what else might DES do. And what they found was that DES also caused mammary cancer, breast cancer in rodents, and especially in rodents that were exposed to DES during early life, during fetal development. Of course, it took several decades for that same effect to eventually be documented in DES daughters, because it takes decades for breast cancer to manifest in human populations. That was a publication that really came out in the early 2000s, and it demonstrated to us how predictive rodents can be for understanding the effects of hormones during early life on the long-term health of the female. So as Anna Maria alluded to, I'm trained in the fields of endocrinology and developmental biology. And really it's my interest in endocrinology that brings me to the study of chemicals in our environment that have hormonal activity. These are referred to as endocrine disrupting chemicals. And I'm particularly interested in chemicals that, like DES, can mimic the actions of estrogens, and understanding what those effects of those chemicals might have on breast health, on breast development, on breast function, and on breast disease. So we live in a hormonally active chemical stew. We're being exposed to chemicals in our everyday lives that can mimic estrogen, that can block the actions of hormones in our body, that can alter the way that hormones are synthesized by endocrine organs in our body, or the way that they're transported through our blood, and also the way that our body processes, metabolizes, and excretes hormones. So again, I'm interested in chemicals that can mimic the actions of estrogens, and those are found in all kinds of products, and we're being exposed to them. I'm literally being exposed to them by being here in my office, because they're released from plastics in the office. They're being released from the electronics in my office and from furniture in my office, in addition to the things that I put on my body this morning in order to keep myself clean, or at least smelling clean. And we're exposed to these chemicals every day, typically at low levels, and often unsuspecting. There's one chemical here, ethinyl estradiol, that's birth control. And most women that take birth control, they know that they're taking that, so they're purposefully altering the function of their endocrine system. But ethinyl estradiol as the contraceptive ingredient is also excreted out into the environment, which means that it pollutes our environment, it pollutes wastewater. So there are non-target species that are being exposed to ethinyl estradiol unsuspecting. The rest of these chemicals are chemicals that my lab has studied, and that we've selected for study because we know from previous work that they mimic the actions of estrogens. And these are also all chemicals where we know that human exposure is occurring, and often at what we think of as low concentrations, but documented across human populations. Today, some of the data that I'm going to show you are going to come from studies of one of these chemicals. But I don't really want to make a big deal about which specific chemical I'm talking about. You'll see it on the slide and I'll reference it. But that's often because it's the best example that I want to give you. Really, I want you to think about all of these chemicals as sort of falling into that bucket of chemicals that interferes with the way that estrogens act in our body. And my model system that I like to use is the mouse. The mouse is a great model because we can control what the mouse is exposed to. I like to tell my students that humans are the most non-compliant patients we could choose, or the most non-compliant subjects. So I work with rodents because they don't have any choice. We get to choose who's exposed, and we can keep a group that's unexposed to be able to compare them. The other great benefit from the mouse is that the mouse has 5 pairs of mammary glands that run along the ventral surface of the animal. And this means that from one animal, we can actually collect a lot of different tissues, all mammary gland tissues, but some to look at changes in gene expression, some to look at changes in the epigenome, some to look at changes in morphology. some to look at changes in histopathology, and they all come from the same animal. And when I think about how the mammary gland develops over time, I like to use the metaphor of a tree. So prior to puberty, the mammary gland has a very small epithelial ductal tree. The epithelium is the functional part of the mammary gland that will eventually make milk. And it's also the tissue where cancers most likely arise. Prior to puberty, it looks like a little twig, a little stump. But at puberty, we see this massive reorganization of the mammary gland. And we see these bulb-like structures, which are highly proliferative cells that are going to help push that ductal tree through the surrounding fat pad. By the time the animal reaches adulthood, that tree looks like a wintertime tree. There's mostly branches here. And it will stay mostly branches, a wintertime appearance, until the female reaches pregnancy, at which point we'll see what look like little leaves or flowers that are budding on the tree, and then they'll start to fill up that tree during the lactational period. At the end of lactation, after the females produce milk and the pups stop drinking milk because they get teeth so they can eat real food, and mama doesn't want them chewing on her anymore, so she will wean them, or in some cases we take the pups away, so it's an unnatural weaning. Then the mammary gland goes through a process called involution, where it regresses, not quite to the pre-pregnant state, but it'll look more like a fall time tree where the leaves are falling off the tree. So I'm going to show you the most amazing thing about the mouse mammary gland is that we can remove the entire mammary gland and put the entire mammary gland on a slide. So what you're going to see is the entire mammary gland that's been placed on a slide. It's been fixed onto that slide, and then it's been stained so that we can see these various different kinds of structures that I've just demonstrated here. So this is what the mammary gland looks like prior to puberty. It's a very small looking tree, not terribly interesting. This bigger branch down here, the trunk of the tree, is the nipple. This is what the mammary gland looks like at puberty. So it's grown a tremendous amount. This prepubertal tree can really fit into a very small part of this mammary gland once it's reached puberty. And this is a close-up view of those bulb-like structures that I was talking about. And those bulb-like structures are full of cells that are proliferating. They're also chewing their way through the surrounding fat network. And the important thing is that these structures, because they're highly proliferative, are also very sensitive to environmental agents like carcinogens or radiation, which makes this a stage of life that can be very susceptible to environmental insults. This is the adult tree. You can see that it's very thin, long branches, not much going on here, not terribly interesting. This is the pregnant animal. So you can see that there's starting to be those flowers or leaves on the tree. And here's a little bit of a close-up so you can see that those are developing. And then here's the lactational mammary gland. And it is full and it is plump. And here's a close-up where you can see those structures that are the milk-producing structures. And so they kind of look like grapes on a vine. And this structure here is the duct. These cells out here will produce the milk. And there's actually cells on the outer parts of these grape-like structures that will squeeze those pockets and allow the milk to go back up through this duct like a pipe to the nursing infant. So I'm going to show you these beautiful pictures of humans because I think after a while you're going to get tired of looking at mice, but just remember that mouse is the model system that we use in my lab. And the first question that we really wanted to have was thinking about how DES was used as a drug to block nursing, to block lactation. We are interested in whether environmental chemicals that have some of the same molecular mechanisms of acting like estrogens, do those also affect nursing in some way? So this is that lactating mammary gland. And now you're an expert in what a lactating mammary gland should look like, because I already showed you one. It really should be this nice, thick carpet-like structure, like you see in the controls here. And you can see, even with an untrained eye, that you can kind of see through this mammary gland from the BPS exposed females, that there are fewer of those structures. And when we evaluated them histopathologically, all these purple dots are those cells that are producing milk, those the functional part of the mammary gland. there were fewer of them in those BPS treated mothers. And importantly, I haven't given you all the details, but these, we refer to them as low and high doses, but they're actually human relevant doses that we're looking at in these studies. And these are moms that were only exposed during the pregnancy and lactational period. So they're responding and their mammary gland is responding during that period of exposure. And it did have effects on the pups. Not only did we see a restricted growth and size of the pups, we also saw changes in the rate at which pups reach really important developmental milestones. So I'm guessing most of you have not seen too many baby mice, but they're born with their eyes closed and their ears are actually closed. And their ears normally open up at about 7 days of age, and their eyes normally open up at about 14 days of age. And you can see that in the controls, 90% of the animals have their eyes open on postnatal day 14. But the animals that came from litters where the mama had been administered BPS during pregnancy and lactation, they were less likely to have their eyes open at that typical stage of postnatal day 14. What we also saw is we can take mama away. We would take her away for about 10 minutes. When we put her back in the cage at day 14, the pups cannot yet feed themselves. They don't have teeth yet, so they're still dependent on the mama, but their eyes should be open, their ears are open. So as soon as we put her back into the cage, they pursue her for milk. You're gonna see that here. I've just returned the mama to the cage, and this is what the babies look like. And I can imagine this mama is super annoyed. She gets no time to herself. These pups are trying very hard to latch on to her. And look at, they're so cute, the little wiggly bodies as they're trying to nurse from her. What we saw is that the BPS exposed pups were less likely to pursue their mother for milk. This may be because they can't see her, but normally they would pursue her just even based on smell and sound. So the pups are smaller, they're not meeting the right developmental stage that they would be at, and yet they're not pursuing their mother for nutrients. The mother's mammary gland is less functional. It's producing less milk. And what we saw was an adaptation in the mother's behavior. Because her mammary gland is less functional, she actually spends more time on the nest. We call these the helicopter moms, right? So normally, as the pups get older and more independent, the white bar, which is the control animal, spends less time nursing. So they give their pups more independence. Even though the pups can't feed themselves yet, mama spends more time away from the babies. That can't happen in the BPS exposed animals. They have to hang out because they're not making enough milk. Their mammary glands are less functional, so they have to nurse them for longer in order to make up for that deficit. We also wanted to know whether exposure to these environmental chemicals that mimic estrogens would also affect other aspects of maternal behavior. There's some evidence from human studies that women who have extremely severe maladaptive maternal behaviors have small genetic changes in their estrogen receptor, which might change how they respond to hormones. And we wanted to know if we've altered hormonal behavior in some ways, hormonal activity, by exposing these females to estrogenic chemicals. Would it affect other aspects of maternal behavior? Now, try not to anthropomorphize these too much. Again, I'm going to show you mice, and you're going to see some things where you're going to think, eek, if a human treated their baby that way, that would be terrible. But they're mice. They pick up their babies by the faces sometimes, and it doesn't hurt them. So don't be worried about that part. One of the cool things that we can do is, again, we separate the mom and the babies for a set period of time, usually for 10 minutes. We take the babies out of the nest that mama has carefully placed them in, and we sprinkle them throughout the cage. And then we put mama back in the cage on the nest. In this case, the nest is up here in this corner. And what you're going to see is that when we return the mama to the cage, she, if she's a good mother, will go and she will check out the pups right away, and then she'll start retrieving them to the cage. So I'm going to show that to you here. So here, this mama, she sees her nest is empty. Oh, she right away touches that baby. She moves it a little bit, but she hasn't carried it back to the nest. So she's trying. Nope, not success yet. Hold, wait for it, wait for it. Here we go. Nope, not yet, not yet, not yet. Come on, mama. There we go. She brings that pup back to the nest. So we're recording all of this so we can keep track of how long does it take for her to interact with a baby? How long does it take for her to retrieve a pup to the nest? How long does it take for her to retrieve each pup to the nest? And then how long does it take for her to retrieve all of the pups to the nest? At this stage, you can see we call these pinkies. These animals not only can't feed themselves, they can't hear anything, their ears are sealed shut, they can't see anything, their eyes are sealed shut. They also can't maintain their own body temperature. So mom really does need to retrieve them to the nest for their own survival. Just so you're not worried, we only give her 10 minutes to do this. And at the end of 10 minutes, we retrieve any pups to the nest that she hasn't. And we cover them up really nicely so that she'll take care of them. What we saw was that in the BPS treated mamas, it took them longer to touch their own babies, and it took them longer to bring their pups back to the nest, especially that first pup retrieval. And so here's an example of a mother who did a great job. She didn't just return them to the nest, she's also covered them up. She's like, please researchers, leave my babies alone. And here's an example of a mama who did not retrieve her pups to the nest, and in fact, she's scattered her nest all over the place. So we help her at the end of that experiment. We also saw this very strange behavior in some of the mothers. So in this example, the nest is way back here. She doesn't want us to spend too much time poking or prodding at the nest. And what you're gonna see is this mom is gonna leave the nest. She's gonna come to the front of the cage here, and she's going to circle and then do this little ballerina spin, and she's carrying her own tail back to the nest. She's gonna do this over and over and over again. And this is an example of a stereotypy. It's obsessive compulsive like behavior. It's a behavior that has no purpose to it. We saw this in our BPS exposed mice, and this example is an ethinyl estradiol exposed mama. She will do this for hours at a time. It's a behavior with no purpose. It passes right around the time when the pup's ears start to open up. So it's a transient sort of obsessive compulsive like behavior that we see in those mamas. Okay, so our next question was, so we've exposed the mother, we exposed her during pregnancy and lactation. Let's take the babies away, natural weaning or unnatural weaning. Let's stop exposing her. Does she recover? Or is she permanently affected because of those exposures that she experienced during pregnancy and lactation? So again, just that reminder, this is what a non-pregnant adult female mammary gland looks like. It's that wintertime tree. And this is what happens in a normal animal that has had babies. She lactated, and then she underwent that process of involution. So this is the fall time tree. So the leaves are falling off the tree, but it doesn't look like a wintertime tree yet. And in animals that had been exposed to oxybenzone, another estrogenic chemical, when we looked at them 5 weeks after involution, so 5 weeks after their last exposure to that estrogen mimicking chemical, what we saw were appearances that were sort of in between the nulliparous and the parous animal, the never pregnant and the once pregnant animal. All of these animals were pregnant, and yet we seem to have adapted or adjusted in some way the appearance of their mammary gland. This is very important because these appearance changes, they're not just appearance changes. They also change the number of stem cells that are present in that mammary gland. The parity, that process of pregnancy and lactation, changes the number of immune cells in the mammary gland. It alters proliferation in the mammary gland. And all of those changes convey to the female protection from future breast cancer or mammary cancer in the case of the rodent. And so our question is, have we also altered some of that protection that comes from parity in the female? This is our most recent study looking at another environmental chemical. Again, you see the nulliparous female, that wintertime looking tree, the parous female, that fall looking tree. And in females that were exposed to propylparaben, another estrogenic chemical, we have an appearance that's sort of in between these two. So this appears to be perhaps a common effect of exposures to estrogenic chemicals during pregnancy and lactation. So to get at that question of whether or not this change in morphology and the appearance of the mammary gland in females that were exposed during pregnancy and lactation might actually also change their risk for mammary cancer. We used mice that were genetically susceptible to mammary cancer. We either kept them as non-pregnant, as pregnant, but they were exposed to a vehicle as a control, or they were pregnant and they were exposed to oxybenzone. And what we saw was a shift, it's a slight shift, but a shift in in how quickly they got tumors in their mammary glands, where it appears that oxybenzone may be speeding up the time that it takes for those parous animals, those pregnant animals, to develop tumors. And I'm showing you over here some examples because everyone likes to see the weird stuff. Those are some examples of the tumors and the kinds of tumors that we saw based on treatment. We also see in the mammary glands of females that have been exposed to oxybenzone, another one of those estrogenic chemicals, we see an increase in the number of double strand breaks in their DNA, and also an increase in the number of these structures that are referred to as R loops. So R loops are a three-dimensional structure that happens when you have DNA and RNA in the same complex. And when R loops are sort of corrected, they introduce the opportunity for double strand breaks in DNA. And that's a risk factor. Again, every time we break DNA, it's an opportunity for a mutation to be introduced. So this is, again, a risk factor for mammary cancer. What we saw was that exposure to natural estrogen, as well as these two chemicals that are estrogen mimics, increased the number of those R loop structures, and oxybenzone, also known as BP3, increased those number of double strand breaks. Okay, so mamas are affected. We see effects on their maternal behavior. We see effects on their mammary glands. What about their daughters? And in these examples, these are mice that were exposed only during gestation and through their mother's milk. So very early in life. This is about the equivalent of, in a human, it would be the equivalent of embryonic and fetal development, and then exposures until probably about 8 to 10 years of age. What we saw in the exposed daughters were examples of anxiety-like behaviors, hyperactivity, and again, those stereotypies, those behaviors that have really no purpose for the female. I'm going to show you a video that's an example of one of these. So this is a female that was exposed during early life, I think in this case to BPS, and she is an amazing back flipper. She will do this kind of behavior all day long for months and months and months of her life. It keeps her extremely fit. So my kin colleagues that are watching, what we see when we dissect these animals is extreme fitness in them. They are very lean, they have very low body fat, but they're displaying this behavior that is extremely abnormal. It's sort of stressing to us to be able to see. I can't ask her how she feels about this kind of behavior, but you can see in the cage, we provided her with alternative things to keep her occupied, like these are little wooden blocks so that she can chew on them, and nothing distracts her from this this stereotypy, this unnecessary, not productive behavior. We also see changes in the daughters that were exposed to these environmental chemicals when we look at their mammary glands. Some of those effects we see at puberty. I'm showing you the examples of females that were exposed, again, only during early life. And then we follow them all the way through to adulthood. And the adult mammary gland, you're now an expert in this, should look like a wintertime tree. And that's what we see in the controls. In animals that are exposed to this estrogen mimicking chemical, what we start to see are structures that have the appearance of those little buds or flowers that are developing on the tree. And these are even more severe in examples of mice that have been exposed to another estrogenic chemical, BPS. If you showed this mammary gland to a veterinary pathologist, that veterinary pathologist, without knowing anything about it, would say, oh, this is an early pregnant female. That's what the appearance looks like as an early pregnant female, when in fact, this is not a pregnant female. The only thing different between her life experience and her life experience is that she was exposed in early life to this estrogenic chemical. Another really fascinating thing that we saw in these adult animals were these bulb-like structures that I'm showing you here. They're pointed at by arrows. And if you remember, when I was showing you the pictures of the mammary gland during development, I showed you these bulb-like structures, but they're normally only there during puberty. And these are structures that are highly proliferative, and they're very sensitive to environmental agents because they're undergoing all of this remodeling and this proliferation that's happening. So the fact that we have seen these structures in the BPS treated females in adulthood is abnormal in and of itself. When we evaluated these structures histopathologically, what we found is that they are precancerous lesions. The strain of mice that we use don't normally develop all the way to mammary cancer. So this is on the pathway to cancer. And we don't know whether or not these would actually lead to cancer, but it's unlikely in this strain of mice. We also took some of those daughters who were exposed to BPS, again, only during early development, and then we got them pregnant. We wanted to see what happens to them. Are they good mothers? Are they producing enough milk? Are they raising their pups normally? And what we saw was that if you were exposed to BPS during early life, you spend less time on your nest. So you're not a helicopter mom like your mama was. You're a bad mom. So you're not nursing them, you're not spending as much time on them. We also saw infanticide. So this is when mothers attack their own babies and kill them. We saw pups that were bruised, dirty. So mom had bitten them, not the kind of biting that she does to carry them around, which is actually quite a gentle biting, but like a harmful biting. So we saw quite abnormal maternal behavior in these females that had been exposed during early development. I just want to warn you, this is the most severe thing that we documented. So if you don't want to watch this, look away for a minute. This is a video of a mother. She was exposed in early life to a very low dose of bisphenol S. And when we came to her cage, we found her like this. So we did not move her babies around. She had not grouped her babies. They're spread all over the cage. And then again, look away if you don't want to see this. This mother is experiencing those stereotypy behaviors, those abnormal behaviors that have no purpose. In this case, it's circling behavior in the cage, and she is stomping her babies to death. And so under veterinary approval, we euthanize all the babies in cages when mothers have these kinds of behaviors, because this is really abnormal. And she did not nurse these babies. She was not caring for them. Okay. In unpublished work that we have, we've also been studying the lactational mammary gland from those females, not the ones who had the abnormal behavior, because their mammary gland is all messed up because they haven't been feeding their baby. So they've been undergoing that process. They've been weaning their babies way too early. So their mammary glands are already undergoing that process of involution. But in mamas who were acting at least mostly normally, that were feeding their babies, what we saw is that their mammary glands have more of that, that fall time appearance than the summer time appearance. When we looked at whether or not their mammary gland was functional, so were they making enough of the good stuff that you should find in milk, we saw changes in some, but not all, of the components that would be found in milk. So the top graph here on the top left is lactalbumin, which is a really important protein that's found in milk. We also saw changes in the expression of PPAR gamma. That's a gene that's involved in making lipid and packaging up lipid. So our data and the data from many others suggests that estrogenic endocrine disrupting chemicals can alter maternal health and the health of offspring. And actually, there aren't a lot of labs that have looked at the mother in like historically, we've been treating the mother as a vessel. So we use her as a way to study what we find interesting, which are the babies. And I'm a developmental biologist, so I totally get that. The babies are so interesting, and the developing structures and organs are so interesting, and they're also so sensitive. But what we've been learning is that the mom is developing too. We haven't thought about her in that way, but we should be thinking about the fact that when mom gets pregnant, there are massive changes in all kinds of organs of her body, including my favorite, the mammary gland. So I've shown you a little bit about the mother and the effect that we've seen on mothers that are exposed and how we've altered their maternal behavior. We've altered their lactating mammary gland. We've altered their long-term mammary gland health. And I've shown you some of our data from the daughters on how estrogenic endocrine disrupting chemicals can alter mammary gland growth and development, how it alters their ability to lactate, how it alters their ability to act as mothers, and also some of those anxiety-like behaviors. I haven't shown you, but we've also looked at other outcomes related to female reproduction and also the ability of the female to respond normally to hormones, as well as to hormonal challenges. Like what happens if you put those daughters on birth control? Do they respond normally to that birth control? And I also haven't had time here today because it's a whole other story, but the story of DES that I started with, where we know that sons are also affected by DES. So sons who were exposed to DES in the womb were more likely to have reproductive tract abnormalities. They are at increased risk for some reproductive cancers. In our lab, for many years as a mammary gland biologist, we were throwing out the boys. They were not interesting to us because the male mouse doesn't have nipples. So we were not looking at the male mouse mammary gland. What a mistake that was. We're realizing that the male mouse is actually hypersensitive to estrogenic chemicals. And we're one of the only labs in the world that is studying the male mouse mammary gland. Some people might take that a different way. Maybe we shouldn't be doing it, but we find this a fascinating and really sensitive sentinel organ. Okay. For many years, this discussion of endocrine disrupting chemicals was really framed as sort of fringe science. That framing mostly came from industries that were polluting our environment with these estrogenic chemicals. They were really good at suggesting that we don't have enough data in order to be concerned about these chemicals. Number one, that probably wasn't true then. It's definitely not true now. And there are numerous scientific and clinical organizations that have taken a very strong stance because the data on endocrine disrupting chemicals from human populations that have no choice about their exposures, as well as tons of data that have come from laboratory animals, really have been convincing that these are chemicals that pose a danger, that pose a risk to public health. And so organizations like the American Academy of Pediatrics, the Federation of International Gynecologists and Obstetrics, and the Endocrine Society have really taken strong stances on these chemicals as a group. So what should we do about this? So I give lots of science talks, and I give a lot of talks also to lay audiences. And at the end, people are like, tell me what to do. What can I do about this? Number one, this is a great slide that I've used when I speak on behalf of the Endocrine Society. There are some things that you can do. So cleaning our houses in ways where there's a lot of chemicals in dust. So just getting dust out of our house, we eat a lot of dust. That's really gross. When I tell my students that and I tell them how much of dust is actually your skin cells, they get really grossed out. We have a lot of dust and we eat a lot of it, and babies eat a ton of dust. So cleaning up dust is a great, simple, inexpensive way to reduce our exposures. Choosing healthy foods, including fresh foods if you can get them, organic foods when you can afford them, and we all have to make choices. So if it's a food that has a peel on it that you don't eat the peel, choosing organic is less important than if you eat the whole fruit. So berries, if you can afford organic, go for it. Don't use pesticides that you don't need, and have a realization of how many things really are pesticides. If you're spraying ants in your house and the thing that you could do to keep those ants away is to be tidier about it rather than spraying chemicals. My yard is full of poison ivy. I do not spray chemicals on it. You can use, well, everything's a chemical, but you know what I mean. I don't buy chemicals at the hardware store to spray on poison ivy. I use vinegar, salt, and detergent as a way to fight it. It's not perfect, but it helps keep it at bay. And the other answer is actually goats, although I have not yet rented any goats to come eat my poison ivy. But it's also absolutely unfair to put the burden on individuals to say, it's your job to now avoid exposures, because it's not possible. It's more expensive to do that. And also, you can't know what you don't know. And I know a lot about this stuff, and I can't avoid these chemicals. I've had my samples evaluated with biomonitoring by colleagues, and my body's full of these things too, with the best of intentions. So we can't keep telling people, if you just did better, you wouldn't be exposed to these chemicals. That's not true, and it's not fair. So what we need to do is to push regulatory agencies to stop leaving the public at risk. And this is really how I spend about half of my time, is focused on how do I get the EPA, the FDA, the European Food Safety Authority, the European Commission to listen to the data and do something about it. Well, we have a big problem. And the problem is that how we test chemicals for safety for regulatory purposes is irrelevant to how people are actually exposed or the diseases that happen. Here's your quick primer on how we test chemicals for safety. Everything I just told you has nothing to do with how we test chemicals for safety. We force feed chemicals to animals, usually for the lifetime of that animal. It could be a year, it could be 2 years. And then we kill them and we weigh their organs. We weigh their organs. That's how we measure toxicity. That's insane. We identify doses that kill them. We identify doses that show overt signs of toxicity, like their hair is falling out or they're having seizures. Then we calculate what we think is a safe dose based on changes in things like organ weight. We compare that safe dose to human exposure. And if there's a big difference between them, we say, yay, we've done a good job. Humans aren't exposed to dangerous doses. Then along come my colleagues in the environmental epidemiology group. They do studies and they're like, uh-oh, there's an association between that chemical and endometriosis or polycystic ovarian syndrome or infertility. But it's a human who's exposed to safe doses. Well, either the entire field of environmental epidemiology is wrong, no students, that's not true, or the way we're testing chemicals is wrong. And I would argue the way we're testing chemicals is wrong. Here's an example. This is not from my lab. This is studies that have been done and evaluated by regulatory agencies. You have to give rodents these huge doses in order to see changes in things that are measurable outcomes that are considered to be adverse. So this is the dose that has no effect on humans. And this is what humans are actually exposed to. So we say, okay, we're great. We're exposing people at doses that have no effect, except we go look at those babies and there are effects on people. And they've been documented over and over and over again. So something is broken about the way we test chemicals. What I think it is that organ weight is a great way to evaluate, are you toxifying an animal? Right? Are you exposing them to something that is making them very, very sick? And that's important. We don't want to expose people to chemicals that make us very, very sick. But I just showed you all those animals acting weirdly, lactating weirdly, weird growth of the mammary gland. None of them act sick. None of them, with the exception of the mama who stomped her babies to death. We don't have good ways of evaluating whether chemicals cause obesity. We don't have good ways of evaluating whether chemicals cause asthma, thyroid disease, behavioral effects. I mean, we do, we just don't use them when we evaluate chemicals for whether or not they're toxic. We're great at evaluating toxicity. We are still crummy from a regulatory perspective at evaluating disease. The other thing is that we need to demand better from companies and industries that make these chemicals. And you might have bought something that says BPA free on it, right? We all have these water bottles and they're plastered with BPA free. Your cosmetics say paraben free. And we need to start asking, okay, that's great. You took that chemical out that we all agree is a bad chemical. What did you replace it with? Because what we're realizing is they took BPA out and they put BPS in. So they can say, honestly, this is BPA free, but they replaced it with a chemical that looks and acts very much in the same way. We can ask companies to do better, but we can also demand through regulation that they do better. This is an example of a study that was conducted in Japan in the early 2000s. It was looking at college students and their consumption of coffee and tea drinks that were packaged in cans with BPA in them. And what you can see is individuals who reported drinking more of these drinks had more BPA in their urine, which makes sense. Then the company changed the composition of the can coating. So there was still BPA in the can lining, but they put another lining between the BPA and the drink. So the BPA wouldn't leach into the drink anymore. And what happened? Didn't matter how many of those drinks you were drinking, everyone's exposure was lower. So a fairly simple change in a product can change exposures. Again, Did they replace BPA with BPS? I don't know. I don't know the answer to that. So we have to stop with that sort of chemical whack-a-mole. This is an example from a study where they did a huge intervention. So they signed people up. They measured how much BPA in this case was in their body prior to the study. Then they did a huge intervention. They cooked for them. They put them in an isolated space so they knew exactly what people were exposed to. Their exposures came down. They didn't go away, but their exposures came down. Then they sent them back to their everyday lives and their exposures went back up. So that intervention of cooking fresh foods and cooking and being careful about what they were feeding people, their exposures went way down. But there was an oopsie in the study. Sometimes you don't know what you don't know. And this is an intervention study where they found out that some of the spices that were being used by an organic chef had been packaged, in this case, in a phthalate. So during the intervention, they accidentally dosed people with more of the chemical. Again, you can't know what you can't know, which is why we can't tell people just avoid these chemicals. Because if we can't do it when we're shopping to run a study, then we can't expect the average consumer to be able to do that in the grocery store. It's just not possible. The last thing that I wanted to tell you is that I sort of hinted at this earlier, but that idea of endocrine disruptors as sort of fringe science is an argument that was entirely shaped by industry. And in this case, we're not talking about one industry that makes endocrine disruptors. We're talking about the pesticide industry and the plastics industry and the electronics industry. We're talking about a lot of vested interests in keeping people to buy stuff and not thinking about how that stuff is exposing them to chemicals. And what we need to realize is that industry has a whole bunch of tactics that they use in order to convince us that the science is not there yet, that there's nothing to worry about, that this is just these wackadoo scientists who are, tree huggers who are saying this sort of stuff about chemicals and, ooh, we want you to be afraid of chemicals. This is all a way to manufacture doubt. And we recently evaluated the tactics that were used by 5 very distinct organizations or industries that have polluted us, that have exposed us to hazardous chemicals or hazardous products, including sugar. And what we found is that there are 28 unique tactics that are used by those industries in order to shape what we think about products and to lie to us and to manipulate us. Many of these same tactics are being used by the chemical industry about endocrine disrupting chemicals. The last point, truly last point I want to make is that if we don't do something about this, we're going to see effects on several generations. We've already started to document effects on mom, on daughters and sons, and what I didn't have time to show you today is also on grandchildren. But other labs have documented that the effects of endocrine disruptors can go to the 4th or 5th generation, including creatures that have never been exposed. And so we need to start thinking about chemical exposures in terms of sustainability and unsustainability. If we're affecting the mother, we're affecting her offspring, we're potentially affecting multiple generations down the road. This is unsustainable. And we should be thinking not just about our children and our grandchildren, people that we know. We should be thinking 7 generations into the future. So these are the pictures of many of my students over the last 5 or 6 years who've contributed to some of the work that I showed you today, and my long list of collaborators and agencies and groups that have funded the research that I've talked about today. And I'm going to leave you with one of my favorites from Calvin and Hobbes, because Calvin has reached the end of his rope here. He says, "Get your toothbrush, Hobbes, we're out of here. It's an outrage how grown-ups have polluted the earth. I refuse to inherit a spoiled planet. I'm leaving." Really? Where to? You're sometimes a real load to have around. I think sometimes this is how people see me when I show up to dinner parties. They're like, uh-oh, she's going to tell me how poisonous my plastics are. I promise you, I am fun at dinner parties, and I'm happy to answer any of your questions.
Hi, Laura. I really enjoyed your presentation. Thanks so much. So I suspect that toxics can also be inhaled. Is that correct?
Yeah, absolutely.
Okay.
Yeah, air pollution is a serious issue. Air pollutants, many of them can be endocrine disrupting chemicals, and the effect of air pollution, not just on respiratory health, but on Alzheimer's disease. It's a wide-reaching impact of air pollutants. Absolutely.
So the residents that are living around the Piney wastewater reservoir in Florida, the one that breached, they were susceptible just because that wastewater was evaporating.
Yeah, absolutely. I mean, they're going to have multiple susceptibilities there. And Mary, you also bring up a point that I didn't have time to make in my talk, which is that we have biological vulnerabilities, but we also have these exposure vulnerabilities, right? We put polluting industries in areas where people are the most vulnerable, the least able to advocate for themselves. When we document these chemicals in people's bodies, they're not evenly distributed across socioeconomic status. They're not evenly distributed across race and ethnicity or age groups. So people get a double whammy.
Okay, thank you.
Thank you,
I really
Mary.
enjoyed your presentation.
Thank you.
So Laura, in the chat, Lindy Sebeko wanted to recognize and thank you for that comment about the inequities related to where these exposures exist. And I think that's something that we in public health need to have a sort of responsibility for, to advocate for those who can't advocate. So thank you for having that vocal voice there and representing us in so many of those different venues.
Yeah, well, and Lindy, I think, could also speak to this idea of like providing people with organic fruits and vegetables, which is a great intervention that most people don't have access to, right? And it's not just that people don't know whether or not it will have an impact on their lives, but people have to make real choices in the grocery store. And as a nutritionist, I can see Lindy's already smiling. You don't want to tell people don't eat fruits and vegetables. Like, we want them to eat fruits and vegetables. So clearly that's a good choice. But we also want to advocate for people who can't afford the fruits and vegetables that are the healthiest.
I think particularly if we're surrounded by communities that live in food deserts, or food swamps, as I like to call them, fast food swamps. So yeah, you've got a point.
Yeah, and that issue of food deserts and food swamps, people are eating stuff we don't even necessarily really consider food, and then it's full of this packaging. So, right, so people who eat packaged food also consume a lot more phthalates, which are chemicals that disrupt estrogens, but also androgens, so sort of more, we think of them as more male hormones. And the consumption of those foods has been documented to affect things like fertility. So, part of it is the food, and part of it is what that food is packaged in, and the fact that it's packaged in that stuff. It also flowed through, like pipes and chutes and conveyor belts and things like that, and it picked up all kinds of chemicals along the way. I saw another couple things in the chat. Hi, Shresti. It's nice to see my students here. She says, you mentioned that toxicity is measured by weighing the organs after the mice die. Can you explain how that works? Why would you do such a thing? So first of all, I should clarify, the mice are not dying a natural death. I know that's like sad and hard to talk about for people who don't work on animals, but those are they're planned euthanasia. And so then you take out the organs and you weigh them. I know that makes no sense. And in fact, one of my mentors and collaborators, a great endocrinologist, Dr. Tom Zeller, uses this metaphor of, it's like taking your car to the mechanic and having the mechanic, and you're like, it's making this weird sound. It goes, wow, 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 wow. And you leave and the mechanic takes out the engine and weighs it. Like, would you take your car back to that mechanic? You would not. And so it's a good way, because you'll see changes in the weight of the liver or the weight of the kidneys if you're really disrupting the way that those organs are functioning. But no one goes to the doctor and says to the doctor, I don't feel well. And the doctor says, lay on the table, I'm going to weigh your spleen. Like, that's not a thing. That doesn't happen. So we're good at measuring toxicity, a change in the weight of a liver. We're bad at evaluating disease, from a regulatory perspective. I'm good at it, I promise you. But what we do when we're evaluating chemicals for the EPA or the FDA is just not disease oriented. And that's just, it's kind of a mess. I see Liz, and you put a question in the chat, so I'll let you ask that question because you got your hand raised. And then after Liz, we'll go to Jody, who's also one of my students. So exciting.
Cool. Well, I'll be fast. Laura, such a great talk. Thank you so much. So my question is specifically about how good the data is for other windows of vulnerability for breast tissue related to endocrine disrupting chemicals and breast cancer. So puberty and pregnancy in particular.
So in humans, the data are fairly crummy, I would say, because we've been pretty bad at evaluating exposures during those periods and then waiting the 4 or 5 decades that we need. So we have really good data from DES. We have some good data from PAHs, polyaromatic hydrocarbons. Those are things that are like when you burn organic material. We also very recently have good data from DDT and DDE, which is a DDT metabolite. DDT was a widely sprayed insecticide. It still is in the environment. It's probably in most of our bodies because it builds up in the food chain. And there were some researchers who had blood in the freezer from the 1970s when DDT was widely used, and they followed up with the daughters. So they have measurements from women during pregnancy. They followed up with their daughters and saw that they were at increased risk for breast cancer. The DES story, right? We know that the daughters are at increased risk for breast cancer. Their mothers were also. So if you took DES during pregnancy, you also have an increased risk for breast cancer. Beyond that, most of what we know comes from rodents. Very few people have studied that period of pregnancy and lactation. So we're one of the few labs that are looking at that period of exposure. Another really important period is the menopausal transition. And I'm not really aware of anyone looking at the mammary gland during that period of time. They are looking at other effects like reproductive senescence. Like do you hit the end of your reproductive period earlier? In women, that would be menopause. Mice don't really have menopause. I call it mousepause, but they don't really have one. So you can see like changes in the number of their ovarian follicles. But this is really, I think, a pretty new part of the endocrine disruption field is understanding all of those vulnerable periods, and they'll probably be different for different organs or tissues of the body.
Great. Thanks so much.
Yeah, thanks, Liz. Jody.
Hi, thank you for such a great presentation. I really enjoyed it. I was wondering about like the BPA and why, like, are people not using it because of the consumer pushback, and why is that not happening with the BPS?
Right. It happened with BPA. Really, to me, this is one of those, like, I think the term is a Pyrrhic victory, where it's like a victory of sorts, but then it kind of burns you. A lot of advocacy groups, probably it's more than 10 years ago, they were up in arms about BPA. Imagine that you find out that a chemical that is added purposefully to baby bottles is estrogenic. It became like a symbol for mom and dad groups of like, how dare you put this estrogenic chemical into a plastic that I'm using to feed my baby with? And so they literally, I went to Boston and marched with this giant plastic baby bottle with all of these. There were like 500 postcards crammed into this baby bottle. And so then the chemical industry actually got really tired because there were all of these states and local jurisdictions that were saying, oh, we might ban BPA. So they would have to send someone to come defend BPA. So I went to Connecticut, I went to Maine, I went to Vermont, I went to Suffolk County, New York, I went to Boston, and I saw the same folks from industry, from the chemical industry there saying, BPA is totally fine. These wackadoos, they think that they want babies to starve. They want, it was awful what they would say about us. I'm like, I don't want babies to starve. I just want us to stop exposing them. So that the chemical industry went to the FDA and said, we are abandoning the use of BPA in baby bottles. So FDA did not ban it from baby bottles, but it was abandoned from use in baby bottles. Then it was 2013. I got a phone call from a reporter because a study had just been done that showed that BPS was estrogenic. And he said to me, Laura, I'm not a chemist, but wouldn't you think a chemical called bisphenol S might have the same behavior as a chemical called bisphenol A? And I said, Brian, I'm not a chemist, but yes, right? Like the chemists have this saying, structure dictates function. If you look at the molecules of BPA and BPS, they look very similar. They look like the Batman symbol. Both of those chemicals, they sneak into the estrogen receptor and they act like estrogen in the estrogen receptor. So that removing of BPA from all of these products, it was done because moms and dads, for the most part, got angry about that chemical. The industry was like, fine, we'll stop using it. We'll now sell you stuff, which we can pretend is good for you, but we've replaced it with BPS. And they could even say, we don't know anything about BPS, because at the time we didn't, because they didn't want to study it. Someone like me had to come along and do the hard work. And there's 29 to 50 replacements for BPA. So it's really whack-a-mole, right? You think you've done something with one, you have to move on to the next, and on we go.
I did not know about that. Thank you for explaining.
Yeah, sorry. I told you I'm fun at dinner parties. There might be one more question in, okay. So first of all, there's do I recommend some books? Please stop by my office. I have a lending library for students who want to read more about these issues. The oldest book that is probably the best book, it just hit its 25-year anniversary, is called Our Stolen Future, sorry, Our Stolen Future. It's 25 years old. I recommend it very highly. I'm looking at my shelf here. My colleague just wrote a book called Sicker, Fatter, Poorer, a great book. You also might want to read the book there's a couple books by David Michaels that are really all about the methods that industry has used to lie to us about chemicals like the ones I've told you today, but also the sugar industry, the asbestos industry, the beryllium industry, and on and on and on. And then I'm going to quickly do it because it's 6 o'clock and you want to go home. You're already home. I should go home. In order to make immediate and also long-lasting change, do you have to target both companies and government? Lindsay, great question, and it's nice to see you also. I honestly think that the answer is going to have to be regulation, which I've been spending a long time and we have not moved the needle on very much. I think the real answer is going to be suing them, which is not something that I want to be involved in. It's something that I think consumers are going to have to be involved in. And there's a great story and also a great talk, I think next week, by the author of a book called The Monsanto Papers. That's all about Roundup and glyphosate. Carrie Gillum, I just read the book this weekend. It is amazing. And it tells the story of how the industry, Monsanto, lied and how they are now paying, if we can keep those verdicts big money, ouchie money. And that's how we make industry respond.
Thank you. I'm curious because when Dr. Savitz had given a talk, I remember him talking about being involved in so many sort of like legal things, and it's just so tricky to do that as a scientist. So it would be interesting to read more on that.
I don't want to touch it, but I hope someone like you will do it for me.
I'll read. Thanks.
Well, thank you, Laura. This was a fabulous Dean seminar. We really appreciate it. You can see how many people are actually praising you in the chat room. So good night, everybody. Enjoy the lovely evening, and Laura, safe travels home.
Thank you.